exam grades are posted. Well, to add to that, just so you're aware, there were 9 questions that I dropped. 9 questions that I dropped to make bonus, making bonus points if you got it right. So, just FYI, that if that puts things in more perspective for you as to your grade and where it stood, obviously dropping those and making the bonus assisted everyone across the board. So, seem to have, it was a tough, it's a tough exam, it's 4 chapters, so yes, you know, that's a lot of information in for one exam. All right, not that it gets much better because we're in the middle of a unit that's got a lot of chemistry. Fortunately, when we're looking at the chemistry aspect of this, we're looking at big picture, okay? So you're not going to be asked to memorize any sort of chemical reaction, but be able to talk through the whole process as a whole, versus the details of the chemical structures or anything like that. That's for a biochemistry class for those of you that choose to go that route, okay? We are going to proceed on with this particular module where we're going to start going through cellular respiration and fermentation. Before I go too much further though, just so you're aware, we do have a lecture quiz on Wednesday, which is October 1st. That lecture quiz is going to be on module 5 up to where we, stop off at. If we finish at module 5 entirely today, so I finish this set of slides, then it'll be on module 5. If we haven't got quite to the last, then I won't cover that on the lecture quiz, okay? Because I don't cover on the lecture quiz stuff we haven't actually gone through yet, okay? All right, and then today, of course, you have a lab quiz, and then next week is going to be something similar. We'll have a lab quiz on Monday. The setup will be similar. We'll have a lab quiz on Monday. We'll also have a lab quiz on Wednesday, even though we don't have a lab on Wednesday. And we will have a lecture quiz on Wednesday. So the lecture quiz will be first, we'll lecture for a while, and then at the end of the class we'll do the lab quiz, as well as follow that by the makeup lab quizzes are next Wednesday, okay? So if there's any makeup quizzes, lab quizzes, not lecture ones, but lab quizzes, that from the start of the term, so basically 1 through 7 that you need to make up, that's the time to do it. I won't remind you. I will remind that saying stay behind if you want to make up at one of those quizzes, but other than that, you need to know which ones you need to make up, okay? And I don't know if it necessarily applies for this term or not, but if you earned a 0 for a quiz, right, then you don't get to make it up. If you weren't here for the class and therefore got a 0 on the quiz, and it would be indicated in Canvas by saying missing, right, it'll be flagged as missing, then you can make it up, okay? Makeups, since the question arises every once in a while, makeups cannot be used, you can't take a makeup quiz to bring up a grade that you earned, okay? And then please don't forget that 2 Mondays from now is the lab practical. Okay, it is October 13th is the lab practical. So make sure you're staying up to speed with that and preparing for it. Also, if you are somebody who works at a distance from the school, or has to travel through downtown to get here or something like that, right, on I-4, make sure that you schedule extra time that day if you need to from your workplace or whatever to make sure you're here by 6 o'clock because the lab quiz will be first, okay? I'll probably repeat this again next week, just to remind people, but just start giving you a heads up. Any questions in regards to coming schedule? Yes?
With the lab practical, is it different from our general bio where it's not like us going to each section for like 90 seconds and doing something? Is it more like a test format, like the questions you have on the website?
It is like as a lab practical, is like what you did in general biology. We'll be playing musical chairs around the room, right? And so every time the timer goes off, everyone stands up and goes to the next chair. No chairs will go missing in between though, unlike musical chairs. But yes, you'll get a minute and a half at each station, and there are 3 parts at each. There's an A, B, and C for every station. And so when I'm doing, and the question style is kind of like what the lab quizzes are. That's why my quizzes are set up are not multiple choice. They're set up to kind of mimic what you're going to see on the lab practical. Differences, is that you tend to have, more time, because there's usually more. I give a minute for a question, usually, or a part A and a part B, but still it's usually a little more time than you would, because in the lab practical, it's every question you get, a minute and a half. For the first lab practical, there will also, though, be a written portion of it, okay? The written portion of it so after we play the musical chairs around the room, you'll turn in that answer sheet, and you'll get another sheet, and that's the one which will have a calculation problem, like we have done for exercise 12, right? Like the practice problems on Canvas that you can go through and be testing yourself with, as far as serial dilutions and calculating the final density, cell density for a sample, okay? Because that's it's one thing to ask somebody to do a serial dilution of a couple bottles, it's a whole nother to do a little too much math, right, into one and say, you got a minute and a half to do it. So, that is a second. I've also already informed you guys that you will have that table, like literally the table for the gram staining. That'll be on the written portion also, and you'll be filling in all of the blanks.
But it's not like the one on the lab practical review, because the one on the lab practical has all of the ones that we did.
No, it's the one that specifically for gram staining, because gram staining is the the other ones are important tests too, but gram staining is always done, and we're going to continue to do it throughout the entire term. So in fact, we're doing it today. So you get continued practice with it. So hopefully, by the time that memorization of that chart helps you, recall fast and your activities of performing that staining, you should be have that pretty much set.
Is it in the academic center where they have your practice, and it kind of depends what yours will be like?
I don't have anything to do with that one.
Okay, so yours will be different.
I have not actually seen that one. I don't know who actually sets it up. But that being said, it's on the micro material, right? So I don't know if a full-time individual instructor actually helped set it up or not. I don't know. I tell you right now that micro is unlike general biology, where everything is lab practical is the same across the board to everyone. The comprehensive final exam everyone takes. The only difference would be the exams during the term. I've taught that one too. Those you have flexibility there, but there are certain things that everyone does. For the micro, it's different. Each like if you take my class and for whatever reason you have to take the class again and you go with another instructor, you're going to find that they might have things that they focus on a little bit differently than I do, right? They may go full on swing into the photosynthesis that I don't. It just depends upon the particular instructor and what they choose to do. There's a lot more flexibility with the microbiology. So I set up my own. I set up the questions. As you already noticed, the questions in the back of the lab each of the exercises, I don't really go and ask you guys to answer those, or we don't, you know, depending, some of them we do actually talk about and answer in our discussions, but I don't go back to them because I've already provided you the type of things I expect you to walk away with. And some of those questions, I don't really care for myself, you know, so I won't ever ask them. You know what I mean? Like, so it just, so I don't know what they have there or not, but I'm sure they have slides at least to be looking at examples of the different organisms, like, you know, for that, for exercise 1, we had there's 5 of them of prepared slides, and so, you know, being able to differentiate. But you also have the pictures on Canvas from my, you know, to have at least something to reference also, and then you have the photo atlas also. So, you know, so, but as far as exact questions, I cannot tell you whether it would be much like mine or not. There may, you know, some of them, yeah, I figure some things are pretty standard questions, right? But then there are other areas, like the questions in the back of some of those exercises where I'm like, I wouldn't ask that, you know. As far as organisms are concerned, the 5 organisms in exercise 1 with the prepared slides, I'll expect you guys to know. And that's just the genus, really. And the endospores, the genus, right? Genus and species could be possible, or just genus for the acid fast, because they're very specific. But most of the other organisms I'm not going to ask. Like, I wouldn't pull up and I've seen some instructors who do, like, what organism did, you know, gram-stained look like this? And I'm like, shoot, it's a gram-negative rod. I've worked in the field, definitely, where I'm like, no one would ever, you don't look at a gram-negative, in a microscope and a gram-negative rod and say, oh, that's Escherichia coli. It's not possible, you know? So, and I understand their wording is, what did we do? But I don't think that's really, you know, me, I don't think that's relevant that you rattle off the name in that case. To me, the gram staining is, can you read the gram reaction? Can you give me the size, shape, and arrangement? That type of thing. That's what's more important than being able to spit out, oh, that's, you know, gram-negative rod, and so it must be E. coli, because that's what we used, you know? So it just depends. But so there's only a handful of actual organisms, and they're related because of the tests we only have, you know, Clostridium and Bacillus. Those are our endospore formers. So both of the endospore staining that we did are going to test for that, you know, those genera. And so that's a fair question. Give me one of them, like we did on the quiz. You know, as long as you can rattle off one of them, even if it's the one we use Bacillus, that's great. We have some people, for whatever reason, Clostridium stuck in their mind. That's great. You know, as you go through the reading for, I think module 6, you're gonna get to see actually Clostridium perfringens. There's a little, you know, those inset extra information, a clinical, where they talk about gas gangrene. Showing a nice, nasty looking picture of a foot that's all, you know, bubbled out because of the gas that they produce from that anaerobic organism. So, you know, whatever makes things stick for you, whatever, you know, that would be, some of those I always have, there's multiple, you can, as long as you give me one, you're gonna be okay. So, any other questions?
Can I ask a question that's not related to the stuff that you said? In the modules in the overview, you and I talked a little bit about, there was a test your knowledge link and packet. When I went into,
None of them for this unit are. Go to my questions. I'm trying to pull up, see, those were something that the other instructor had, but for this particular unit, they did like little quiz questions, and they had like, I didn't like the quiz questions. Like they were quiz questions on Canvas. I didn't really care for many of the questions. I didn't, you know, it was kind of more specific. So I'm trying to find, number one, go to the overview for the unit, right? Because most of the stuff, even for last one, it was, you know, if you take those questions, spread them out on a piece of paper, it's, you know, to a module, it still covers. But I'm trying to find a couple extra little.
No, there's not really like, so kind of use the module as like a study guide or a reference sheet as to where we should.
Yeah. But
lot of
I
information.
am looking for some things to add into there.
Okay.
I may or may not be able to find what I'm looking for, or it may, the couple things that I have find might not cover the whole module versus,
Mhm.
it might just be, say, specific. I found one that I kind of liked on, cellular respiration. It was more of a flow chart style. So, and then had a, word bank, and you kind of fill in, just kind of, it's more for representation, and I didn't,
That's
I
nice
saw
for
it,
exercise,
they
like-
forgot to upload it type of thing, so I was gonna do that, but I was debating on, again, it's just for cellular respiration versus, all the other components, so it's not testing knowledge on everything. Again, I would go back to the review questions for the unit. In the unit 2, I have already out there review questions for unit 2.
Yes, so
And
download
those
that and use
are-
that, okay.
Yeah, those are gonna be in a similar fashion.
Okay,
Yeah. But thank you for asking, because I am working towards it, but I will say may or may not be as encompassing. Even though I put worksheets, it's not necessarily gonna be encompassing to all aspects of that module. The unit, the review though does have encompassed. Okay. Any other questions?
I saw on the bonus part, it said it was 80 question, or 80 points.
No, 5 points for the vote.
And then in parentheses after it, it had 80 points. Is that like total 80 points? So like,
No.
I'll show you after class, how about that?
Because
Yeah.
that must be something else.
It looked like you went like totaling them all available or possible, and then in parentheses you put 80.
No, that's, for the,
I thought 80 was only for the lecture.
80's for the lecture quizzes, and the bonus points are placed in the lecture quiz area. I believe that's what it's referred to, where I've got it, so.
I didn't see that, and I already submitted my extra.
Okay. I think it's in the area for, it with the graded area, it's in as far as, I grouped all bonus points with the lecture quiz, the lecture bonus points with the lecture quizzes. So it's probably referring to the 80 points for lecture quizzes.
Yeah, I'll look at it again, maybe I looked at it wrong.
Okay. All right, so, so now today we're gonna talk about, cellular respiration. We, hopefully get to fermentation also. Cellular respiration, this is where we're looking at, taking our most common carbohydrate for energy source, that is glucose, and catabolizing that. Now, glucose can be catabolized by either two mechanisms, cellular respiration or fermentation. Fermentation is an anaerobic process. That means oxygen is not present. That being said, cellular respiration can also be an anaerobic process. There's two types of cellular respiration. There is aerobic cellular respiration and anaerobic cellular respiration. And that's going to really be based upon the final electron acceptor at the end of that process. Okay, so cellular respiration, and we're going to go through these different steps, but cellular respiration consists of glycolysis, where we take our glucose and we're going to break it down, 6 carbon molecule, we're going to basically reorganize and break it into 2, and so that at the end we're going to have 3 carbon molecules. We're going to have 3 carbon molecules, and that would be pyruvate or pyruvic acid. Okay, same thing. In this process, of course, we're going to get a little bit of energy captured, okay? Then that pyruvate, in the presence of oxygen or in some of for some bacteria in an anaerobic environment, that pyruvate can then move into the Krebs cycle. Now, even as you look at this this text that we're using, it refers to it as Krebs cycle, but then some of the figures are referring to it as a citric acid cycle. It's also called tricarboxylic acid cycle. They're all the same thing, okay? So they all mean the same thing. I tend to refer to it as Krebs cycle, okay? So if you hear me saying it, it'll be Krebs cycle. When you're testing on it, it'll be Krebs cycle. Okay, but at any point, if you were to be looking at another text or some other area, and you're going, oh, but this is citric acid, it's the same thing. Don't worry about it. Okay. Now, Krebs cycle in itself is where it is a cycle, so our start product is our end product, and it is recognized by the fact that we get a lot of electron carriers produced from this process. Okay, it is also the process, we do get a little bit of ATP produced, but it is also the process where our glucose molecule, that carbon skeleton that is now 2-3 carbons, has been broken down completely and released as CO2. All right, and then the last step of the cellular respiration process is the electron transport chain with ATP synthase. This process is known as chemoosmosis, where they link the two. What's going on in the electron transport chain, which does not in itself make energy in the form of ATP, okay? But it sets up an environment that acts like a battery, and ATP synthase uses that environment that is set up to then produce ATP. So those two are combined. The electron transport chain actually itself is where all those electron carriers are going to drop off their electrons, and those electrons are going to move down in potential energy through this series of molecules, and then be dropped off with what we call the final electron acceptor. Okay, for aerobic cellular respiration, okay, that final electron acceptor is going to be molecular oxygen. That is O2, so the gas. Okay. For anaerobic cellular respiration, that final electron acceptor is going to be a molecule that has oxygen as part of the molecule, but is not the gas. It is not O2. Okay, so it could be nitrate, nitrite, carbonate, that type of thing. So it's going to have oxygen as part of the molecule. Okay, that's then going to accept those electrons. Okay, so this process, cellular respiration, is our most efficient, and of the two, between aerobic and anaerobic cellular respiration, the most efficient is our aerobic cellular respiration. From one glucose molecule, we can get about 40% of the energy from that molecule, right? 40%, you say, is, if you think about it, and we were to talk about our grades, right, we'd be going, that doesn't sound very good, right? But for the purposes of obtaining energy, this is actually considered very efficient. It's considered, it is actually considered highly efficient, and the energy that we aren't able to capture, that is actually lost to heat, okay, dispersed out into the environment. Anaerobic, we'll look at this later, we'll have a slide on this. Anaerobic cellular respiration can be as efficient as our aerobic cellular respiration, or it might be as inefficient as fermentation. It depends upon the particular organism that is performing that procedure. Now, fermentation is known to be a partial breakdown of glucose. So in fermentation, you'll notice we have glycolysis, but then we don't go any further. We don't go past glycolysis. So glycolysis, we're going to walk away with a net of 2 ATP. That, right, is not a lot of energy that we're going to be able to obtain from that glucose molecule. It drops us down to an efficiency of about maybe 2 to 4%. Okay, pretty significantly less, right? So you say, why would a organism then ferment, right? If you have an option for fermenting and getting about 40% of your energy from that glucose molecule as compared to fermentation, we're getting about 2 to 4%. Why would we go that route? And the reason is that fermentation is an anaerobic process. Oxygen does not have to be present at all, okay? So in some organisms, they will only perform fermentation. Other organisms can alternate back and forth between the two. You, as an organism, actually have cells, your muscle cells, that have the ability to alternate between the two. when you are expending energy, your muscle cells are expending energy, your body's ability to bring the oxygen to that tissue is not, it doesn't have the ability to bring it fast enough, and therefore it switches gears and goes into a fermentation process. You're still being able to get energy from those glucose molecules. It's not going to be as efficient, but what's going to happen is you're going to move through, and you're just going to use more glucose molecules, okay? And for us, of course, with fermentation, we have end products or byproducts that are produced. For fermentation for us and our muscle cells, that'd be lactic acid. But for these microorganisms, it depends upon the particular organism. It could be anywhere between the acids or the alcohols, okay? Where is that coming from? Well, at the end of glycolysis, you have two of these pyruvic acids, and we are going to have those electron carriers. The electron carriers are going to drop the those electrons off with pyruvate itself directly, or with a derivative or breakdown product of pyruvate. Okay, and that's where you're going to get your acids and alcohols produced. All right, so for cellular respiration, this is a picture that kind of visually, and it's not focusing on everything. Okay, so I just found this off the internet, but it kind of visually displays it of in this process. Of course, we're going to be breaking down glucose. This first step is to go through glycolysis. Glycolysis will require that we have an input of energy. We'll be using 2 ATP to get this process going. We will need to use our electron carrier NADH, right? But by the end of glycolysis, we're going to have 2 pyruvate. We will have 4 ATP, but of course you have to pay yourself back. You use 2 ATP, we produce 4 ATP, right? So you're gonna have to pay yourself back, and so that is a net of 2 ATP produced. And then we have the NADH. Our electron carrier now we'll have 2 of those. Electron carriers have picked up electrons. Now, the net 2 ATP that are produced during glycolysis can be used right away by the cell, okay? Because ATP is like cash in the cell. NADH is like savings bonds, okay? You can't go to the grocery store and say, I'm gonna pay for my food, here's my savings bond. They're not gonna accept that as a means of, as a transaction means, okay? However, that's not to say it doesn't have worth. It does have worth. It has worth if you go to a specific location, okay, such as a bank, and say, okay, I am going to exchange this for the cash value, right? So our electron carriers, those NADH and later FADH2, are going to end up going to the electron transport chain. That's where we're going to get ATP from that molecule. All right, now the pyruvate, as I already mentioned, both pyruvates are going to go through the Krebs cycle one at a time, okay? Not as pyruvate directly. Pyruvate is so we have, this is like a 6-carbon skeleton, right? So we got 6 here. Now here we're going to have 2 3-carbon molecules. To get into the Krebs cycle, incorporate into the Krebs cycle, it's going to have to become a 2-carbon molecule. So our first CO2, we have 2 CO2s because we have 2 molecules. So our first CO2 is going to be released. And then it's going to catch to coenzyme A. Okay, that is going to then enter into the Krebs cycle. Now, with the Krebs cycle, we get a lot of this produced. We get, this is an FADH2. It's hard to read it from here because they've made it small, but we do get FADH2 as an electron carrier also produced from the Krebs cycle. We will get ATP. So in this case, we're going to get ATP produced. Also, we're going to talk about this in more detail later. So from Krebs cycle, so those will be able to be used. That ATP from Krebs cycle is actually going to be produced from an intermediary. We're actually going to produce GTP first and then ATP. Okay. So by the end of Krebs cycle, do they have it? Yes, they have it. By the end of Krebs cycle, we've released all of our CO2 from our glucose molecule. So all 6 CO2 have been released. So we no longer have any carbons from the original carbon skeleton. Okay, we started off with 6, we released 6. So as we exit that carbon cycle, we started with the start product becomes the end product, and we walk away with some ATP, but a lot of electron carriers. The electron carriers are going to go to the electron transport chain, and this is where we're going to see production of our ATP. Okay. Now, to go into more detail, and then of course these pictures, some of these pictures like this one is coming directly from your text. So to go into glycolysis, okay, starting off with glucose. We have, glycolysis is going to be split into 2 phases. We have our energy investment phase and our energy output or payoff phase, right? To get the ball rolling, to get the process going, we actually have to use some energy in the form of ATP. Okay, so we're going to need to use 2 ATP to get this process going. So glucose is going to have its first phosphate attached to that glucose molecule by an enzyme called hexokinase, and we're going to get glucose 6-phosphate produced. So now we have a 6-carbon molecule with 1 phosphate attached. As we move through this process, we're going to get another phosphate attached and a reorganization. Okay, so we have glucose 1,6 or fructose 1,6 phosphate that is then going to be, which is a 6-carbon molecule that's going to be split into 2. Okay, the 2 molecules are each going to have 3 carbon skeletons with a phosphate attached. Okay, one of those molecules is going to be one is going to be glyceraldehyde 3-phosphate, which is what you see in this picture here. Okay, now you do see 2 glyceraldehyde 3-phosphates. Originally, one of them is glyceraldehyde 3-phosphate. The other one is dihydroxyacetone phosphate, which is an isomer of glyceraldehyde 3-phosphate. So we'll have an isomerase. Remember, our enzymes are categorized. Isomerase is an enzyme that changes the configuration structurally so that it is a different molecule. So that in the end, we're going to have 2 glyceraldehyde 3-phosphates. At that point, we're going to move into the payoff phase. And during the payoff phase, both of those 3 carbon molecules are going to go through configuration changes and chemical reactions where we're going to have electrons pulled off, okay, added to NAD plus to give us NADH. We will also produce ATP, okay, so we'll get per each of these, we're going to get 2. So if we've got 2 here, right? In our payoff, we get 4 ATP, and we're going to have 2 NAD plus H. Sorry, NAD H. NAD plus is going to take the electrons, and we'll get 2 NADH. So from this 4, don't forget we had to subtract 2 ATP because we used it. So this is plus, right? So if we're looking at what is our payout to this whole process, this is where we have to end up saying, okay, we made 4 ATP, we have to subtract 2 ATP, therefore our net gain, right, is 2 ATP. I'm missing an A. All right, we have, our net gain is 2 ATP. So we've got, per glucose molecule, we get 2 ATP, we get 2 NADH, and we have our 2 pyruvate that we're going to walk away with. Okay? In this process, we have substrate level phosphorylation. Substrate level phosphorylation occurred when we had ATP transferring a phosphate to an organic molecule here. It is also happening here with the substrate level phosphorylation to produce ATP. Okay. So this net gain of 2 ATP is through substrate level phosphorylation. All right. Oh, whoa. Where am I here? That we just did this one. All right. So then that pyruvate needs to go to the next stage. Oh, no, before I leave here, just one thing. Where is glycolysis occurring? It doesn't matter whether we're talking about a prokaryotic cell or a eukaryotic cell. Glycolysis is occurring in the cytoplasm. Okay, so regardless of the type of cell, it occurs in the cytoplasm. All right, so the next is those pyruvate. They have 3 carbons. We have 2 molecules of pyruvate, 3 carbon molecule that is now ready to move forward. We want to continue to break it down. We want to introduce it into the Krebs cycle. Okay, as such, it is going to enter in as a 2 carbon molecule. Therefore, we're going to have our first decarboxylation. Okay, carboxylation is to remove a CO2. So we're going to have our first decarboxylation process. And we are going to remove our CO2, one CO2, and then that 2 carbon molecule will be attached to coenzyme A to give us acetyl-CoA. In the configuration of acetyl-CoA, this can now enter into Krebs cycle. Now, this portion is sometimes called an intermediate step. Okay, but we still group it with Krebs cycle. You'll notice as we went through this process of pyruvic acid to acetyl-CoA, we actually got another electron carrier. Okay, so at this point for NADH, we've got, so far we've got 2, right? 2 just from this step of going 3 carbon to 2 carbon so it can enter into our Krebs cycle. Okay, we've also released our first CO2. The Krebs cycle in a prokaryotic cell is going to occur in the cytoplasm. In a eukaryotic cell, it's occurring in the matrix of the mitochondria, so the mitochondrial matrix. Okay, so in a eukaryotic organism, this step here is going to occur as that molecule transfers into the mitochondria. So there is a separate, the Krebs cycle is happening at a separate location in a eukaryotic cell than in a prokaryotic cell. All right. Acetyl-CoA, we're going to have those 2 carbons attached to that coenzyme A. It's going to get transferred over to the start and end product of the Krebs cycle, which is oxaloacetate. Okay, so it'll get added to oxaloacetate, and then we're going to see a series of, nice thing is we have no details here, thank goodness. So you don't have to worry about getting clogged up with a whole bunch of chemical structures. We're going to have a series of chemical reactions occur. During the first half of the cycle, the job is to be releasing the CO2. Okay, as well as in the process, we're going to get NADH produced. Okay, and then at some point about halfway through, we are going to get our ATP. It is generally through first adding a phosphate group to GDP to make ATP, GTP, GDP to make GTP, okay, and then the phosphate gets transferred from GTP to ADP to give us ATP. Okay, so it'll go along the lines of, let's take away this for a moment. We're gonna get GDP getting to GTP, and then we're gonna have this GTP, we're gonna have a DP going to a TP, and this will end up going back over to GTP. So we will get, in this reaction, our ATP. So in the end, and this is, so in the end, 1 per cycle, we'll get one ATP, but we have 2 pyruvics going through this cycle, so the cycle's gonna happen twice. So we're gonna get a total of 2 ATP. Now, in the continued process of getting back to the original starting molecule, we're gonna get more NADH and FADH. So in our total, we will get a total of 3 NADH, right, plus our original 2. And then we will get, we have to times this, I'm sorry, we have to times this by 2, because it's showing one cycle, so there we go. And then we'll also get the electron carrier FADH2, and we're gonna get a total 2, 1 per each cycle. So NADH, this is our electron carrier. This is an electron carrier. In the Krebs cycle, we get NADH just like we did in glycolysis. Okay, so we get NADH will come from both glycolysis and the Krebs cycle. But in the Krebs cycle, we'll get an additional, and that type of electron carrier, and that's that FADH2. That's only coming from the Krebs cycle. We will, by the end of the Krebs cycle, we will also have released 6 CO2. Right, so what we have released, that will have been sent out into the environment. Okay, so our original will have, our original 2 3-carbon molecules will now have been broken down completely. Okay. Between glycolysis and Krebs cycle, so far we've come up with a total of 4 ATP. 2 net from glycolysis, and we've got 2 from Krebs cycle. That being said, what we have a lot of are the electron carriers, those savings bonds. So in comes the next step, which would be the electron transport chain. Okay, the electron transport chain is made up of electron carriers. These electron carriers, though, are anchored in a membrane. Unlike NAD plus and FAD that are free within the cytoplasm in the mitochondrial matrix, the electron carriers, electron transport chain are locked into that membrane. Those electron carriers that consist of, there's 4 categories of these carriers. That we have plasma protein, ubiquinone, metal-containing proteins, and our cytochromes. And as these, so all of those electron carriers that we obtain through glycolysis and Krebs cycle, all those electrons that we've been pulling off of the molecules in order to break it down so that we now have no more of the original 6-carbon sugar left. All of those electrons are attached to those electron carriers, NADH and FADH2. We need to get them off, right? Because then those electron carriers need to then go pick up more. They have a job. Their job is just to go, like a taxi, go pick up and then drive somewhere, drop off. So they can go and pick up more and drop off. Okay, so where do they drop them off to? They drop them off to the electron transport chain. The electron transport chain, those four groups of electron carriers, are going to step down the potential energy in little steps, like a staircase. Okay, we have the flavin protein would be at the top of the staircase, and then we have our ubiquinone and metal bearing ions, then we have several cytochromes. And at the very bottom here, we have our final electron acceptor. Okay, so our electrons are going to drop off on this with these electron carriers and bounce down in potential energy until eventually they get dropped off with that final electron acceptor. Okay, that is so that we can slowly release energy, and in that slow releasing of energy process, we can try to acquire, okay, that energy, lock it into our system, trap that energy. Now, as those electrons are bouncing, right, so it's going to just go bounce down this series of electron carriers. We have a couple places where electrons are going to be hydrogen ions are going to be transported across that membrane as this occurs. So hydrogen ions are going to move across the membrane at here. 3 different locations. These are indicated here, here, and here. Flavin protein is where one hydrogen ion pump is located. When the electrons pass by that electron carrier, it's going to force those hydrogen ions to move across that membrane. Okay, it'll then go to ubiquinone, and then we have 2 of the cytochromes. We have a cytochrome A, B, and C. 2 of them will have transport those hydrogen ion transports. So as electrons pass those, hydrogen ions get pushed to the other side. And as a result, what you get is a high concentration of hydrogen ions. Oh, not the hashtags there. Hold on. Hydrogen ions on one side, right? Then as compared to the other. So one side is more positively charged than the other side. That's the battery system. These 2 that is set up, right? We have a lot of positive charge over here, a lot of negative charge over here, and we've got a membrane that separates the 2. This is the purpose of the electron transport chain, is to set up this environment. Okay? Now, the last, though, is to look at that final electron carrier. So the electrons have gone through each of these, bouncing down the potential energy, till they eventually reach the final electron acceptor. If we're talking about aerobic cellular respiration, that electron acceptor is molecular oxygen, O2. Okay, and when it's accepted, it also takes in hydrogen ions, again, decreasing the hydrogen ions on this side, and we get water produced. If it is anaerobic cellular respiration, you'll have something like nitrate, so like something like NO3. It has oxygen just in that molecule to accept those electrons. Why oxygen, by the way? Do you remember the importance of oxygen in the chemistry side, in the module 2? What do we know about oxygen? That molecule, that atom. It is highly electronegative, right? So it's going to be drawing those electrons. So molecular, if it's oxygen, it's going to direct, and we're going to get water. If it's another molecule, for instance, in an anaerobic cellular respiration, we still have oxygen there. It's going to pull those electrons. Okay, and you'll get a different product from that. Not water, but still, we don't. So that's really the difference between anaerobic cellular respiration versus aerobic cellular respiration. Aerobic is oxygen, molecular oxygen, O2, specifically. All right. So now we have an environment set up. wait, before I go too far. Before I go too far. We have the environment set up, assuming, this here, okay, this was NAD, plus those electrons, right? So the electrons that were attached to NADH drop off at the top of the staircase. Where does FADH2 drop off its electrons? FADH2 drops off its electrons at this location, okay? So it's 2 electrons plus FAD. Those electrons get dropped off here and they bounce. The final electron sector being whatever it is, you know, whether it's oxygen or for aerobic or anaerobic, something else, right? Notice that FADH2 drops off its electrons not at the top of the staircase, but somewhere in between, right? And by dropping it off at this point, which is actually ubiquinone in the middle bearing protein, excuse me, by dropping it off here, notice it misses one of the hydrogen ion pumps. Okay, so we get more of that chemical gradient set up from NADH when it drops off its electrons than we do with FADH2 when it drops off its, right? For NADH, we're going to get an increase of the 3 points of hydrogen ions being pumped across. Just FYI, hydrogen ions, when they're pumped across, are pumped across in sets of 2, just like the electrons move in sets of 2. Okay, so I have for NADH, I have 6 hydrogen ions pumped across total at the 3 different points. For FADH2, I'll have 4 from the 2 different points. Okay, because FADH doesn't pump across as many hydrogen ions, do you think we're going to get as much ATP from that? No. Okay, we're not. We're going to get less ATP produced from FADH2 molecule as compared to that we're going to get from NADH. Okay, now, where's my, erase this portion here. Okay, so now we bring in ATP synthase, right? We still have our higher energy, our higher hydrogen ion concentration over on one side as compared to the other, right, of that membrane. This is the electron transport chain portion. Now we're going to look at ATP synthase. ATP synthase is going to take advantage of that hydrogen ion concentration difference. Okay, hydrogen ions are positively charged. We're going to have a negative charge on the other side. Those hydrogen ions are going to want to cross if they have a pathway. Well, guess who provides the pathway? ATP synthase. ATP synthase consists of 2 regions. We have, or domains for this particular protein. The domain that's sitting inserted into the membrane, okay, that's going to create a channel for those hydrogen ions to pass through to get to the other side. And then we have a catalytic region. That's the portion that's going to be involved in oxidative phosphorylation. It's going to take inorganic phosphate and attach it to ADP to give us ATP. Okay, so as hydrogen ions go rushing towards that negatively charged area and pass through ATP synthase, it's going to actually turn like a turbine, and that energy then is going to be what is used to attach that inorganic phosphate to ADP to give us ATP. Okay, and it basically equates to, for every pump that was crossed, hydrogen ions crossed, you're going to get 1 ATP. So if for NADH, 2 hydrogen ions, 2 hydrogen ions, 2 hydrogen ions for a total of 6. 2 come in, we get 1 ATP. Next 2 come in, we get another ATP. Next comes in, we get another 2, another ATP. Okay, so for each NADH, we're going to get 3 ATP produced. For each FADH2, because we lose one of those hydrogen ion pump regions, we're only gonna get 2. Okay? All right. That ATP synthase is actually performing is what we were, again, oxidative phosphorylation. It's the first time we're seeing. The other 2 steps, glycolysis and Krebs cycle, did substrate level phosphorylation. Here is where we're seeing oxidative phosphorylation. All right. From the NADH and FADH2 coming from glycolysis and from the Krebs cycle, we're going to get dropping off their electrons. For those that are coming from per 1 glucose molecule, we're going to get 34 ATP produced. We had already 2 from ATP from glycolysis. We had 2 coming from Krebs cycle for a total of 38 ATP total produced per glucose molecule. And again, that's about a 40% efficiency of capturing the energy in that glucose molecule. Okay. This event, you'll notice what they're showing here specifically is showing you where this event occurs in a bacterial cell, because you'll notice here they talk about outside the cytoplasmic membrane, so outside the membrane, and inside the membrane. For bacteria, this event is going to occur across the cell membrane. For eukaryotic cells, this event is occurring across the inner membrane of the mitochondria. So this is occurring inside the mitochondria for a eukaryotic cell. For a prokaryotic cell like bacteria, it's occurring across the only membrane it has, which is the cell membrane. Okay, so location here is different as to where that activity is actually occurring. All right, so from this process then, we have glycolysis, right? The flow of those carbon skeletons is we're going to have a 6-carbon glucose molecule that's broken down into 2 3-carbon molecules. We'll end with pyruvate. In this process, we will have 2 NADH that are produced. This is not energy immediate. These are our savings bonds, right, that need to go and be, those electrons are still being held by this electron carrier to be deposited at the electron transport chain. And then we will have 2 ATP produced by substrate level phosphorylation. Okay? Stop here right now for this figure. They have it set up a little differently than I'm used to. This is coming from your textbook. Okay? Now, as far as I'm concerned, these 2 are actually kind of linked, right? In order for pyruvate and those carbons to get into the Krebs cycle, we have to have that transition phase. The transition phase in a eukaryotic cell is going to occur as that molecule moves into the mitochondrial matrix and then where the Krebs cycle occurs. For a prokaryotic cell, this is just going to continue in the cytoplasm. But the transition is that we have our 3-carbon pyruvates, those 2 3-carbon pyruvates, becoming 2 acetyl-CoA. Okay. So it'll lose, which is a 2-carbon molecule, so it'll have lost the first of the CO2 from each of those 3-carbon molecules. In this process, we will get NADH produced, 1 per each pyruvate. Then once we're in the Krebs cycle, that acetyl-CoA is going to, those 2 carbons are going to be placed onto a molecule and then go through that cycle, during which we're going to release CO2. So the total of CO2 for the 2 acetyl-CoA is going to be 4. So at this point now we've released 2 here, another 4 here, for a total of 6 CO2 just through this Krebs cycle process. The original 6 has been released. So this is CO2, it's going up into the environment. Okay? so as a gas. In Krebs cycle, of course, we have our electron carriers, 6 NADH, 2 FADH2 for the per glucose molecule, plus we produce 2 ATP. So between glycolysis and the Krebs cycle, that combo, we get a total of 4 AT produced. Not a lot, okay? Not a lot of energy immediately available for use by the cell. However, that being said, we have all of these electron carriers, which can then move to the let's see, electron transport chain and ATP synthase, right? So it moves to this portion. And this is where we are going to have all those molecules, those electron carriers come into play to give us ATP, so that by the end of the process, we'll have 38 ATP. You may, on occasion, hear people will say 36 for eukaryotic cells. That has to do with the fact that sometimes when we're transitioning into the mitochondria, some of those eukaryotic cells will have to expend energy to get it into the mitochondria. So they'll use an ATP per molecule of pyruvate, and so it drops the number a little bit, right? But 36, 38, you know, if you're in that level, you're looking at the right answer, okay? All right, so where does that put us in relation to then fermentation? Now, I don't 100%, again, pulled off the internet, don't 100% like this picture, only because of the fact that they're going off of a cell that can transition between, and this is kind of talking about a cell that can transition between aerobic cellular respiration and fermentation. Okay, so they have, when oxygen is present, you have this happening, what we just covered. Glucose going to glycolysis, we have our intermediate step to get to the Krebs cycle, and then the electron transport chain, with then water being a product as the electrons are accepted by O2, right? So we get water. Okay, I don't disagree, that is the aerobic cellular respiration. Okay, for a cell that can alternate between aerobic and anaerobic and fermentation, excuse me, so aerobic respiration and fermentation, when oxygen is not present, that cell is going to go into fermentation. Glycolysis does not require, is a chemical pathway that is independent of oxygen. It requires no oxygen. And therefore, glycolysis can proceed. Glucose can be partially digested through the glycolysis process, giving us pyruvate. But you then have these electron carriers, these NADH, that are holding onto electrons. They gotta get rid of them. Because again, electron carriers, as taxis, have to drop these electrons off somewhere. What they do in this case is they drop it off onto an organic molecule. That organic molecule happens to be pyruvate, or a breakdown product of pyruvate. And in that process, you get your acids or your alcohols produced. Okay, so there's nothing wrong with the figure in this regard as far as how it's being presented, except for this. There is such a thing as an anaerobic process. So O2 is not present, where we see glycolysis, and we see prep steps, we see Krebs cycle, etc. Yeah. And then the electron transport chain, right? But in this case, it's not going to be in the mitochondria. This is going to be across the cell membrane. and it's not gonna be O2, it's going to be that oxygen bearing molecule to give us whatever the end product is. It's going to accept those electrons, those 2 electrons, to give us, it depends upon what that oxygen bearing molecule is as to what our end product will be. It won't be water, it'll be something else. This though, here, this is by prokaryotic. We definitely see this in prokaryotic cells. Okay, this is aerobic cellular respiration. So this whole side is cellular respiration, and this side is fermentation. Okay, so that's the only difference. The only reason I don't fully agree with this particular slide, there's focusing on a eukaryotic organism, and this happening in the mitochondria, and us having oxygen as the final electron acceptor. There is an alternative, but it's limited to a smaller group of organisms, right, that can do that particular activity. All right, so let's see. So now we're gonna go into fermentation, and its comparison with aerobic cellular respiration, anaerobic cellular respiration, and fermentation. I would never ask you about the organisms, so I'm not really interested in that. The difference between aerobic and anaerobic, here's your final electron acceptor, right? And we have molecular oxygen here, nitrate, sulfate, various of our other molecules that have oxygen present in them. Okay, that are going to take in those, to attract those electrons. As compared to fermentation, where are our electrons going to go? It's going to go to an organic molecule, usually pyruvate. Okay? In this process, we already just talked about aerobic cellular respiration, where we ended up with a total of 38. Okay? Anaerobic cellular respiration, it can vary. It can be as efficient, almost as efficient as aerobic cellular respiration, or it might be almost as bad as fermentation as far as how much energy we can trap. The bulk of that is actually based upon the electron transport chain. In fact, remember the different electron carriers and electron transport chain? This group here are the cytochromes. And some of your bacteria don't have all of them. Okay, so if they don't have, for instance, one of them that has a hydrogen ion pump related to it, then right off the bat, you've lost a hydrogen ion pump. So you're already reducing the amount you're gonna get per NADH and F8H2. Okay, we actually, when characterizing bacteria, that's one of the tests that they will do is, do they have cytochrome A? Do they have cytochrome, B? Do they have C, etc. In order to determine, how much efficiency they're getting, and it's a unique characteristic of that particular organism when it plays in the bigger picture, right? And then fermentation, since it's just glycolysis, all you're getting is those net 2 ATP. NADH, did you get two NADH? Absolutely, you got 2 NADH. They carried electrons. That's a savings bond. A savings bond that has to be cashed in with the electron transport chain, but there is no electron transport chain with fermentation. So the electrons can't be cashed in. So what they can do is they get dumped off. They get dumped off with an organic molecule, and that's where you get your acids and your alcohols produced. Okay? All right, so For fermentation, glycolysis is the first thing that's gonna happen. The glucose molecule is going to be partially oxidized, right, to produce through glycolysis, and you'll have that 6 carbon molecule at the beginning of glycolysis. You'll end up with 2 3-carbon molecules of pyruvate. You'll end up with 2 ATP, net ATP, and you'll end up with 2 NADH. The NADH have to get rid of those electrons. So where do they go? It depends upon the organism. One thing that they can do is take those electrons, drop them off with pyruvate to produce lactic acid. Okay, that's what our muscle cells do. There are quite a few bacteria that do the same thing, right? They produce lactic acid when they drop off those electrons. Another option, and that's a type of acid fermentation. There's many different acids that could be produced: formic acid, succinic acid, acetic acid, right? It would depend upon the particular organism, and do they drop it off with pyruvate, or do they break down pyruvate a little bit first and then drop it off? Okay, that again depends upon the particular organism. Alcohol fermentation. Alcohol fermentation is one in which pyruvic acid first is broken down. This is a 3-carbon molecule. It's broken down into a 2-carbon molecule with CO2, of course, being released, right? And then that 2-carbon molecule is what receives those electrons to produce that ethanol. Okay, that is a form of alcohol fermentation. Alcoholic products are going to have then that alcohol, plus you also see the carbonation, that CO2. Okay, it's a natural product of the process. All right, so this is, again, only 2 examples of potential outcomes. It really depends upon what breakdown products and where those which organic molecule those electrons land with, as to what the products are. There is no Krebs cycle here with fermentation. There's no electron transport chain. There's no ATP synthase. As a result, we only see substrate level phosphorylation here. No oxidative phosphorylation. Oxidative phosphorylation, again, is linked with the chemoosmosis, electron transport chain, and ATP synthase. That's where we see oxidative phosphorylation. We're not going to see that with fermentation. Some of the products of fermentation. You can see from the list we have multiple outcomes that we can get with fermentation. You have some organisms and some and then like our muscle cells that are referred to as homolactic fermenters. That means with fermenting, they only produce lactic acid. We have others that are heterolactic fermenters. They produce lactic acid as well as a whole bunch of other products. Okay, so so here we've got a mixed acids: acetic acid, formic acid, lactic acids there too, succinic acid. Even in this mixed acid, we've got some alcohol produced, CO2 released. So this is a broad range of different end products that these particular organisms can do. We have our alcohols, and again, alcohols and acids that are going to be possible, right? Products that are produced by these. We're looking at alcohols, you're looking at solvents, gasoline alternatives. If you're looking at Saccharomyces, it's the one that we use for beer and bread making. Okay, some of our wines can be made by certain wines can be made by Enterobacter or Klebsiella. Others are made through Saccharomyces. And then we've got butter products, sauerkraut, yogurt, cheese, vinegar, some of your pharmaceuticals and cosmetics, as well as your cheeses, right? If you're looking at Swiss cheese, the big holes in Swiss cheese are due to the production of the CO2, that gas that gets trapped in that milk product as it's fermenting that cheese. Okay, so a lot of different options for the fermentation, right, the products that could happen. And it depends upon the particular organism. Again, this is where, as we go to the unknowns project down the road, right, we do a series of biochemical tests, okay? And at this point in time, you don't need to know these biochemical tests, right? You're not gonna be tested on this at this point in time, but down the road with the unknowns, you'll actually perform them. At that time, you would need to know them, okay? So we'll go through some of them at that point in time. But some of the tests that you'll perform are things such as carbohydrate fermentation, where we'll have test tubes with just one carbohydrate. So we'll have the 5 different test tubes, each one with their own different carbohydrate. So you're gonna look at and kind of start building a profile for your particular organism. Do they ferment this? Do they ferment that? Do they ferment the other, right? If they ferment it, do they ferment it? This is no fermentation. These two are indicating fermentation due to the color change. This is what we call a differential test. So we're able to see a difference between the test results, right? In this case, this one this organism ferments it but doesn't produce gas. This organism ferments it, but you see that little bubble that's trapped in that upside down tube? Okay, that indicates that bubble was produced because when fermenting, the bacteria produce gas, and of course gas rises and it gets caught in the bubble. So now we know this organism ferments whatever the sugar is, and it ferments it with gas production. Okay, we have other tests such as the methyl red test here that looks at mixed acid production. So this has got glucose, this media has glucose in it. Okay, so does it ferment it doesn't ferment glucose. This one does ferment glucose. I'm sorry, this one is the negative. I'm sorry, I switched. I should probably have the reverse. This is the negative result. This is the positive test result. This says it produces mixed acids. When it ferments that glucose, this says it does not produce mixed acids when it ferments. Okay, and then this test, the Voges Pasteur test, is one in which we're looking at acetylene production. Okay, so we're doing, again, glucose fermentation, but what type of product do we produce? Okay, so multiple tests that what we can look at, and this is just looking at fermentation, not even as we proceed forward, you're gonna see other tests that will look at, say, specific enzymes, right? And when we do these multiple biochemical tests, we produce a profile. That profile is unique for that organism and allows us then to characterize the organism. And if we have an unknown, it allows us to identify our unknown based upon those test results. I'm going to stop here today because I think that's way more for you to, gnaw on. So what we have left that won't be covered on the quiz coming on Wednesday would be the catabolism of lipids and our proteins. I think that's the remaining things that we have left on this particular module. No, just